Thanks, Ernst. So SMC is for Statistical Methodology and Consulting. And I worked in the pharmacometrics group, I think 10 years ago when I entered Novartis, but then I moved to the stats department. But anyway, I'm located in Basel and very honored to give this StanCon keynote lecture here to you. So let's get started. I assume that you are not from biostatistics, so I will give you a bit of a broader introduction into the topic, basically. The following is my opinion, not the opinion or any views of my employer, Novartis. So let's jump right into it. So the pharmaceutical industry I'm working for is aiming to provide safe and effective treatments for patients, and that's happening in a certain setting of certain. So the involved parties are patients with a disease, obviously, medical doctors treating these patients, healthcare providers like hospitals where trials are being run, basically, sponsors like Novartis or other pharmaceutical companies, and health authorities like the FDA or EMA, who have the mandate to protect public health, basically. They review data from research programs from pharma companies and authorize treatments for the market. Then there's also another piece, which is public health governance, like institutes like NICE in the UK or IQWiC in Germany. These are involved in determining drug pricing, basically, which is also very important to make sure that actually access to new treatments is possible. And it's a highly regulated environment. It's an international setting. So by now, the International Council for Harmonization has been founded a few decades ago. And what they do is they regulate and give guidances out for just about everything what we do. How you collect data, how you ask for informed consent from patients, how you store data, how you handle data. But actually, I emphasize now data. There's a lot more than just data, obviously, in such a clinical trial. And the ICH authors guidances, which are then regionally implemented as law. So not everything happens not exactly the same everywhere in the world, but according to these guidances, everything is harmonized, which makes this more efficient and ensures that patients are safe and trials are run according to some common standards. Okay, drug development. What is drug development? Drug development itself, to me, actually starts later than these 2 points. We would first have a discovery, typically of a molecule, which would then become a pill eventually. But the treatment nowadays can be actually very different. For example, you can extract cells from patients, treat those cells and re-inject those cells. These are CAR T treatments nowadays. So what's the treatment nowadays is very diverse. Then you would have a phase of preclinical development. Mostly this would involve animal testing. And then we enter clinical development, which means treatments are being given to human beings for the first time. Before it's authorized and can be sold, it has to go through the development of typically 3 phases, phase 1, 2, and 3. Phase 1 would be, we're asking the question, is the drug safe and does it actually do anything, hopefully useful? In phase 2, we would ask, how should we dose? Should we give it daily, weekly, twice a day, or in what strength should we give it, and so on and so on? Should we make the dose dependent also on weight or things like that, basically? And phase 3, and these first 2 phases is a learning phase, so decisions taken there are at the risk of the sponsor. So that's why health authorities wouldn't be as much involved in the decision process there as when it comes to phase 3, when the data really is collected to answer the question, does it work and is a new treatment safe for use? And the data, mainly from the phase 3, but of course the entire data, is then submitted to the health authorities and they review if the treatment is safe for use and efficacious and if the benefits risks are in relation to one or so that it makes sense to authorize the treatment. obviously, if you have a life-threatening treatment, then you would be okay with some safety issues, versus if that's not the case, then you would be more rigorous on safety. There could be another phase 4, which are optional post-approval studies, collecting possibly additional data, so you may not have gotten yet the sufficient exposure to patients to the drug, so that the authority would like to have maybe more safety data being collected. Or phase 4 trials could also be run to basically have a say on drug pricing, because you want to do more comparisons against other treatments already available. And yeah, that's where obviously this is a big ongoing decision process, and quantitative approaches do support this all along. Here I list now things I've been involved on. So yes, I started at Novartis in the pharmacometrics department, and pharmacometrics is basically interested in building longitudinal models on the drug concentration and their resulting effect on the patient. And this is usually expressed using ordinary differential equations, and that's also where I've been working on a lot and providing code to Stan, actually, so that ODEs can be solved in Stan. So the models being used are population pharmacokinetic models, which ask the question what a patient's body does to the drug. So you give to a patient a pill of a certain strength. He will then absorb that pill. He will distribute the pill in his body. He will metabolize the pill, and he will eliminate the pill. So that's the ADME process, which I've just described. And that's what population PK is about to describe. So you know then, as a function of when you dose a patient, what concentration you have at any time point after the dosing in the bloodstream of the patient. And these are so-called population models, which just refers to the fact that all these parameters which exist in these models are patient-specific, and we fit these using hierarchical approaches, basically. Then we have population pharmacodynamics. So now we know the concentration. So what does now the drug actually do to the patient? So yeah, what does the drug now do to the patient, which could be the effect of the drug in terms of efficacy or safety, basically. And again, we use population models here. Now, in more statistical modeling, we deal with diverse endpoints, like continuous endpoints, change from baseline of some score, binary endpoints, responder type of analysis. So you define that someone is a responder at some fixed time point after baseline. If something changes in a good way, obviously, that's the way it's typically defined by convention. Or you look at time to first events or time to recurrent events problems, for example. What makes these things typically more fun and interesting from a statistical perspective are intercurrent events leading to missing data. So you have your baseline where you randomized patients to either treatment or control, but then you measure at 12 weeks, and patients in the meantime may drop out of your trial. They may even switch the treatment arm, that is sometimes also allowed. And then the question is, what do you estimate here, and what's your target estimate, how it's called, and which method you use, and all of that, of course, needs to be well thought through, in particular for a phase 3, as you will pre-specify it. What I've also worked on safety models for guidance of adaptive trials in oncology, for which I actually also authored an R package, OncoBase 2, and I'd defer you to the vignette of that R package if you want to read more details on that. I've also worked on combining average data with individual level patient data in the context of nonlinear models. So drug development typically happens not in isolation of the world. Very often, actually, there are already existing treatment on the market. Maybe other competitors will also develop drugs in the same indication. So there's other data basically out there. The issue usually we have is that only the data of the trials which we collected and executed ourselves, we would have available in the form of individual level patient data. And those you can then use to create nonlinear models, which will allow you to infer a lot more usually interesting properties and learn about your drug. However, the competitor, and typically in the literature, one would only receive and get summary level data over time, basically, so not the IPD data. And putting these types of models together is something I've worked on with colleagues, basically. Today, I'd like to concentrate on the use of historical, typically control data, as I think this is first of all, this is a very relevant problem for early phase drug development, and it's also a very nice Bayesian application, I'd say so. The use of historical control data is a case study on our new website, which we've launched earlier this year. Applied Modeling in Drug Development. This is the URL and the QR code source code, so it's a fully public open source website with introductory material on a wide set of problems from drug development and how you approach these using BRMS, basically. And this material is joint work of Novartis Associates. So these case studies were really on the desk of my colleagues, basically, and we have put them in the form of simulated data onto the website. And jointly with Paul, who kindly agreed to work with us, and Paul put this into the form of a workshop. And so this entire material here on the website is the result of 3 years of 3 times a workshop at Novartis, which was very fun to do with jointly with Paul. And I'm also glad that Novartis was okay to now make this material public and open source. So these are really, if you want to read about what biostatistics is, and then have a look at these case studies, all the data is simulated data is contained all the source code is there. Just last month, after having gone public this year, we actually also gave a course at the JSM conference, so last month in August, and the conference workshop material is also available for download. Okay, the use of historical control data. Applications of using historical control data in drug development, there are many. As you can see, I list some I've worked on. So first of all, we are talking here about the use of historical control summary data, not IPD data. So summaries of like how many responders you had or things like that, summaries. And the goal here is to reduce the sample size in your control group while maintaining statistical power when comparing an active treatment arm, for which you wouldn't have typically any any historical information, to the control group, because the control group has been seen and observed in clinical trials already a few times before. You could also use historical control information to get an idea of, to better plan hand, basically, to define what you have as a true effect assumption, or learn about the variability in your population, or calculate in advance the probability of success. And there are a number of other applications which are very interesting and relevant, but we're not going to touch them today. So, but before we now continue, let's first consider how we would classically basically design a simple 2-arm trial. So a trial means we want to test out our new treatment and compare it against a control typically being placebo, giving no treatment. Although you would of course give to patients still a pill, it's just that it doesn't contain any active treatment, because the trial would obviously be blinded. So patients would enter your trial, then you would at baseline assign a treatment randomly to be either active or control, and then this information would be blinded to the patient and to the doctor, and also to the sponsors, obviously, up until database lock. And then you would wait maybe for 12 weeks, and after 12 weeks you make another assessment, And then you compare that to the baseline, and then you can find out if your treatment improved outcomes or not in comparison to control. And you would do that by first defining your type 1 error rate for rejecting the no effect hypothesis H0, which states that the effect parameter denoted with theta p and theta t is exactly the same in both groups. Then you would choose your sample size in both groups on a true effect assumption. The alternative, that there's truly a delta between the difference between the two groups in this parameter. And you would assume a certain desired type 2 error rate beta, and this will tell you the sample size you need to have in each group. And you would quickly find out that the one-to-one randomization ratio would have highest efficiency for such a two-arm trial. And then in the frequentist framework, which would be the standard way to analyze such data, is you are guaranteed that the type 1 error rate to reject the null hypothesis is controlled at any alpha and at any value for theta p, theta t, and for any sample size n. This is what it means to be controlled. And this all goes out of the window once we now go Bayesian. And that's why it's very important to be very careful in documenting these designs. So we now need to define, or what we want to do, we need to define an informative prior in parametric form, and that's also very important. We want to be specific and write down in our statistical analysis plan, in our SAP, what's the prior, and we don't want to dump there an MCMC sample, basically. And then we also need to document statistical design properties. Okay. So let's look at an example, which is in the R-Best-R package, which I wrote, which facilitates this. We'll get to that. A double-blinded proof of concept trial, which is placebo-controlled trial, and the results of this trial have been published in The Lancet in 2013. So this is an actual trial which happened. And the endpoint here is a binary ASAS 20. So this is ASAS is a continuous scoring for those patients, and the 20, I'm not sure anymore, but I think it refers to a 20% improvement in that score at week 6. So improvement would be more, higher rates, higher response rates are better. At the time of planning the study, there was a big amount of data already available. In this indication, a number of trials have been run. And in this specific patient population, basically, 8 studies have been conducted prior to this trial, including overall 513 patients. And usually that data would be ignored because we would want to run it frequentist typically. And this is where this trial, one of the first trials, did actually leverage this historical control data, ran a fully Bayesian design with the success criteria defined to be that we want to have 95% assurance that the the treatment response rate exceeds the one in the placebo group. For the placebo group, so-called MAP prior, we'll get to that, was used, which was derived from the historical data, which you see on the right here, where you see the forest plot for the 8 different studies and the reported 95% credible intervals. And below you see the mean estimate from a meta-analysis and the MAP prior, which we get to as said. So this was summarized to be a beta prior at the time. Nowadays one would do it with a mixture beta, but we'll get to that. And on active, an uninformative prior, which makes it actually a bit harder to have higher response rates. It's why it's not beta 1,1 was used. The randomization ratio was chosen to be very aggressive, of 4:1. So 24 patients were put on active. So you would see, regularly you would compare the 24 patients on control, but instead 18 patients were dropped and instead replaced with the informative MAP prior here. So the map prior, just to look at it, you see the response rate is around 25%, and basically in the range of 10 to 45, 50% response rate, which is what you would get from the historical data. Okay, so what's now the meta-analytic predictive map prior? This approach has been introduced by Beat Neuenschwander in 2010, a former Novartis colleague of mine. And the idea is that you want to borrow from historical data under an exchangeability assumption. So you see here maybe 3 different historical data items, capital Y, 1, 2, and 3. We model these using individual control parameters and then say on a common scale these are linked to one another on an exchangeability assumption using typically a normal distribution, but using an exchangeability assumption so that we assume that we have a common population mean beta and a between trial heterogeneity tau. And because we introduce between trial heterogeneity, we do discount data, meaning we start with 513 patients worth of data in the historical data, but what this corresponds to as what you derive from a prior will be a lot less, and you will see that. And now the map approach now, meta-analytic predictive prior now works through the nice property of the hierarchical model that is generative. So knowing something about items, some items in my data set, I can predict. So I basically I'm interested in the predictive of the mean of my new trial, which the hierarchical model will tell me. So there are obviously a few prerequisites. Namely, we essentially have to look at the same endpoint here. Otherwise, it wouldn't make sense, obviously. It needs to be similar treatment procedures need to be also similar. We need to have the same patient eligibility criteria. So we don't allow just any patient to come into our trials, but we ensure that patients are have a certain disease status, so to say. They must be adults, and so on and so on. So usually a number of criteria which need to, of course, align when you compare the historical data to what you plan for yourself, basically. And you need to have still similar patient population characteristics. So roughly the baseline covariate should match. However, you don't get to pick your historical data. So it should be similar. And that's also why this concept of between trial heterogeneity is important. Basically, you try it by, so to say, by not having everything perfectly aligned with heterogeneity with variance. So you discount, that's why you discount the historical data. There are also alternative techniques available, like the power prior or the commensurate prior, which both work in a similar, basically, way. They discount the historical data. That's what they basically do. They just work differently. Okay, so the map approach can be implemented with the R-based R package, which I've wrote. R-based stands for the R-Bayesian Evidence Synthesis Tools, and it's also, this is now, I think, since 2015 open source. So this was done, also made public in order to get the map approach being used more in the biostatistical community, and that's actually happening nowadays. So it's designed to help you with the key steps, although the first one is a bit outside of the realm of software, obviously, to assess the historical data for its relevance. And there you would need to think of how heterogeneous is your data, which becomes relevant when you then set up your prior for this model. So do you only use phase 3 data, for example? Do you use phase 3, phase 2, and maybe some phase 1 data, which are typically less controlled than you can reflect this in your prior on tau, basically. Then we would run this meta-analysis, which is just actually a standard meta-analysis with a prediction, that's map, in order to obtain this informative prior in parametric form. The meta-analytic model would be run using MCMC to get the posterior. For that, Stan is being used. It's a very simple model But I can tell you people throw super uninformative priors on it, so even then Stan has to sweat a little bit. So it's good that we have the warnings from Stan as to complaining when it's too uninformative. Usually that's what happens. Then as a second step, and that's also very important, we have now MCMC, and now we need to approximate it. We want to write down explicitly what's the prior. Writing down an MCMC sample in a sub isn't going to work. So we need to approximate it, and we use parametric mixtures approximation of the posterior sample, for which in RBEST we have very efficient implementation of the expectation maximization. Efficient for this type of problem, I have to say. And then we evaluate frequentist properties like type 1 errors and type 2 errors, which look very different than what I've alluded to before. So let's look at it in more detail. The generalized meta-analytic predictive model. Capital Y is the control group summary data for capital H historical studies. So we allocate for each historical data item one control parameter, theta H, and put forward a likelihood, F, in the case of binary data, of course, a binomial likelihood. And we do the same for the new trial, obviously. And another way, by the way, to think about this prior is to just declare the data of the new trial to be missing. That's another way to view it, and it's totally basically leading to the same results. But now, so far, these are just a bunch of parameters, so we need to learn something. So we put forward an exchangeability assumption, so we transform to an unconstrained scale using the logits function for binary data. Then on the logout scale, and on the logout scale, we put forward that all the different parameters come from a common normal distribution with a population mean and a between trial heterogeneity tau. And we, of course, also support the other canonical likelihoods and functions in RBEST. The nitty-gritty parameter is really tau, because this controls between trial heterogeneity, and the key limit cases are tau approaching 0, or very small values, basically, meaning there is no heterogeneity in the data, meaning these parameters, they cannot vary much around this beta, so we will basically, if it approaches 0, we will pool the data, leading to an unbound use of the historical data. If tau approaches infinity, or meaning we have super large heterogeneity, we will stratify everything. The heterogeneity is so large that knowing something from the past doesn't help you at all for saying something about the future. So you will not use your historical data, basically. You will not have a prior in your hand in that case. So running now the map analysis in R, on the right you see the data set, which is included in the RBEST R package. Here you see the RBEST code, and as this package has been designed to solve this problem, it's obviously relatively straightforward. What you don't see is that RBEST will force you and not let you get away with not specifying a prior on tau. So, and here it's a very conservative actually one. Because if you simulate a half normal 0, 1 on the logit scale, that's very, very conservative. Here's the respective BRMS code, and I'm bringing this up as on the website. We solve this also with BRMS, since BRMS would allow you to do far more flexible modeling, have more random effect structures, because RBEST has only just one random effect on the intercept. Some basic considerations here. So the model with many historical studies, and 8 studies is a lot actually, is largely insensitive to the prior choices, and is, from my view, a very good example for what I would call Bayesian thinking. So we put our trial data into the context of the world, into the context of other data, basically. This is Bayesian thinking, but priors themselves are not really, really important in this case. You can just throw in priors which are known to be super conservative, and the data will take care of the rest, and you're good.
So, Sebastian, what do you mean by conservative here?
Conservative, that's a good. People would love to not put any prior on tau, which they will quickly find out it doesn't work. Conservative meaning, typically people say that I want to allow for huge heterogeneity and don't want to single out certain values. So this is already, I'm telling you this, and I'm using a half normal 0, 1. Maybe for you it doesn't sound very informative, or yeah, but this is would be our standard by now recommendation to use if you have lots of trials and want to be conservative, basically. But actually, if you know that your data is more homogeneous anyway from the start off, like you have phase 3 data or something, using a half normal 1/8 would be something which I would probably then recommend more. But the usual case is actually that we have less than 3 historical trials available. Maybe just 3 or maybe just 2, and in the extreme case, maybe only 1. And in that case, obviously, this analysis immediately becomes very, very Bayesian insofar that the prior which you put up on tau, the prior which you put up there will be seen in what determines the posterior, basically. So the choice of the prior is really relevant in that case. But it's one of the most interesting cases because very often you don't have 8 studies. This is basically the textbook example you use to introduce and motivate and do the approach for the first time. But then, if you are clever about choosing a good prior on tau, then you can actually use it in those cases when you just have 2 studies, for example. However, guess how long it took about to settle on a prior in this domain? More or less than 10 years? More than 10 years it took, basically, is what I would say. So by now, I think there's good agreement in the literature that one should use a half normal prior with some scale S. It's not, in all honesty, the favorite prior I would have chosen. I would have maybe chosen a PC prior or something like that, but it really, it's super valuable that the literature in the field settled on this prior, and this is really what you should never what I would never dismiss, basically, when I run this analysis. So the scale S is suggested to be set in relation to sigma, so it's endpoint specific. There's a comprehensive overview by my colleague Christian Weber from 2021, see 11 years after. And what's also noteworthy is that there is an empirical evaluation of the German IQWiG by Lilienthal in 2023. And the IQWiG, to remind you, they assess the data from sponsors. So they do meta-analysis typically of twin phase 3 studies and want to combine the evidence. And they want to do this with a meta-analytic model, which is very nice that they want to do it in a Bayesian framework. But they just had to learn the hard way that it doesn't work with a frequentist approach, because usually this institute only applies frequentist approaches and I can understand that. I mean, they are an institute which have to set drug prices and they want to make everything as data-driven as possible. But what they did, they reanalyzed their many phase 3 studies they have, their cases, of which they compiled more than 100 for the data set. So they do the meta-analysis of a meta-analysis to find the scale of S, and it's documented in that paper. So I'm very happy actually about the paper. And the priors, when you look at them, they really reflect things are not so heterogeneous. It's really, when I looked at this, I thought, whoa, this is super informative. But I mean, that makes sense. Twin phase 3 studies, it's the same thing being replicated. So now, so far we've just our MCMC sample. Now we want to have our parametric approximation. And we do that with a conjugate mixture priors estimated with EM. Note that I switched from the logit scale to the 0-1 scale here for a problem, because then we can use beta mixtures. Yes?
On the 0, 1, or the half normal prior, do you have any issues when it gets close to 0? Like, don't you see some?
No, this is still when I am on the logit scale. So this is still when I'm on the logit scale. So exchangeability assumptions, random effects models on the logit scale. So there's no issue with that. But still, then I have my MCMC sample of the logits, and then I transfer it back to the 0-1 scale because of when I work with beta mixtures, basically. Because then I can leverage analytical results for posterior updating beta mixtures. And you see, if you just use one beta mixture, it doesn't really approximate well the MCMC sample, but I mean, the beta hasn't been made to approximate these types of results of analyses. But instead, we just use more, which is better. as you can see in the top right. Using 2 beta mixtures is already very accurate approximation. And using 3, it's, I would guess at that point, already good enough. And these are then parametric approximations which you can write into your protocol, basically. Now, we do one more thing, which is to robustify the analysis. So we add a non-informative mixture component to it, which is there to account for the possibility that there could be prior data conflict. If there's a conflict between the map prior and the data, then Bayes will be super stubborn in updating, basically, as it will form a compromise between prior and data. And the effect of this robustification is to deal with that, essentially. And technically, it's equivalent to Bayesian model averaging between using the map model, which puts forward the exchangeability assumption, and no model, basically. Now we have our prior. Now we want to know what's it worth. And that's the concept of the prior effective sample size is very helpful there. Conceptually, for a simple case, assume you have a normal endpoint with known sigma. The information of a normal prior is basically given by its standard error in the usual equation as we know well. And sampling standard deviation divided by the square root of the sample size you have collected to estimate a mean. And you can invert and you get your effective sample size. For mixtures, it's a bit more complicated and involved. There are various methods out there. For example, moment matching, you would match the moments to a simple beta in this case. But this is very conservative, meaning giving you very low effective sample sizes. Morita would be based on the curvature at the mode. It's very liberal, so how peaky is your prior is what it's saying. And the method which Beat and myself introduced in 2020, which is ELIA, which is the expected local information ratio, it's basically it's based on the average curvature, and this is the only method which has one of the cool properties, which is called predictively consistent, which you see at the bottom. Happy to go into more details at the end if we have more time. And if you want to spare 45 minutes, me presenting on ESS, this is a YouTube link which you can watch. So statistical design considerations. Now the informative map is then there to enable unequal randomization by substituting sample size of the control by the prior information while maintaining trial power. Doing this discounting bit already reduces a lot the effective sample size. Doing robustification reduces it even further. As we see when we calculate the ESS with the ELIA method of the map prior, which is 36. Remember, we started with 513 patients. Now we robustified, and then we are at 25. Doesn't sound like a lot, but remember, classically, we would actually enroll 24 versus 24. So having a prior of effective sample size 25 is a lot. Not using any control patients is a bad idea, actually, because then you lose randomization and blinding. So this is what we would not do. So looking now at the type 1 error, the frequency for go, whenever the rates are exactly the same in both groups. I compare here the flat design, which is unfair, which uses 24 versus 6 and uses an uninformative beta 1,1 prior. But it's nicely controlled. You see at the bottom of the type 1 error, basically. The informative map prior, you see, has a huge type 1 error here on the right of the plot. And the robust version of it is an intermediate between the two. So what happens here on the right, we say we have a response rate of 80% in the control group, which is supposed to be the true, but my prior is pulling towards 25%, so biasing in this direction towards smaller. So then artificially creating a big difference between the two. So you would conclude very often that you actually have a success when the reality is there's no success. But there's something heavily flawed with this plot. Maybe people have seen it or noticed it. It goes up to 1. It goes even beyond 50%. But if you remember from the historical data, we should actually look at this x-axis. So it's not anymore as drastic, actually, because we've never seen a response rate in the control group going beyond 45, 50%. So it's not as drastic, basically. So type 1 error is what we would call the risk of using such an approach, and the power frequency for go is here you see the gain. So here I've added a fourth curve, the black one, which is sort of say the reference design, using an uninformative beta 1 prior and comparing 24 with 24. And here, as a function of the difference between the two groups, we see that, yeah, at 35%, we have our 80% roughly power, which is what you would aim for. And you see with a fully informative prior, we are even better than that. But nowadays, and this was run back then, but nowadays one would probably choose the robust design, which is basically as good as the 1-to-1 randomized 24 with 24, but with 18 patients less on control. And yeah, the flat for 24 versus 6 just doesn't give you what you would like to see. Okay, but we can make it more fun. We can actually throw in covariates. So map with covariates. Very often we would have baseline adjusted, for example, chain score models. These are very common in phase 3. And very often we would have prior knowledge from earlier studies. So yeah, covariates make your life more complicated in this case setting. And this is another case study from our website. Let's assume we have the following data constellation. We have a hypothetical phase 3 evaluating some treatment for a rare disease. Capital Y is a primary endpoint denoting the mean change of a patient-reported outcome from follow-up, which would be after 12 months after baseline. We have Z, the treatment indicator, X1 and X2 indicators for the age group for young and old. So the reference age group would be the medium age group. And C base, in this type of analysis, you would always want to adjust for the baseline score in the PRO. And this is then now the standard baseline PRO score. A basic phase 3 model, analysis model would look like this, using the IPD data in the individual level patient data. And here, yeah, I'm just writing down a normal Gaussian model with a linear predictor using those covariates which just introduced. Now let's assume we have two historical studies on control, a natural disease history study and a control arm of a randomized study evaluating another treatment. Here we now only have the summaries reported, so we get the change in the mean and the standard error for the different age groups, and we don't even have this age group information for the second study. Okay, this is now not anymore in the realm, or I will solve this now with BRMS. And writing it down with BRMS, it's relatively straightforward. The only special thing is that you have to condition on the reported standard errors in the data, basically. And I'm adding here also now the between study heterogeneity on the intercept. And priors are being set basically in reference to the sampling standard deviation as we've seen it in the historical data. Okay. Now we want our map prior, which means we want to have the posterior of the parameters, but BRMS itself is tailored and built to give you posterior predictions of the linear predictor or of the mean, which will already compound the different parameters for you, but that's what we don't want actually. So we need to do a bit of trickery basically and manipulations to get at the end the posterior of the different parameters. Okay, still MCMC sample, but we want to document things in parametric form. And here is where our best can give you a multivariate normal mixture fitted to this posterior sample and summarizing it for you in parametric form. Of course, I mean, going multivariate normal means it's a few more parameters to write down, but still you can do that. Now, and also what's very important to be aware of that these mixture distributions, as we add more mixtures, components will get more and more accurate actually. And it's only then more work to write up those parametric forms in the protocol. But if you want to have it more accurate, you can just add more mixture components. But obviously we should also check if we actually did a good job here. And RBAS also gives you these neat figures comparing for you the pairwise plots of the MCMC density to the fitted analytic density. Okay, fine. And we see we have strong correlations in the posterior, which is expected given this type of model. Let's now try out the approach and use some simulated data of as we would expect the data to read out. This is now, of course, IPD data. And there's one more thing which I haven't really talked about, but basically I have integrated out, said that this is a new study, and then instructed BRMS to simulate the random effects level basically here. So we have now the uncertainty due to the between trial heterogeneity included in the intercept at this stage. And this is now the final BRMS model. This is stand code, so some stand code obviously is due. This is now the basic phase 3 model, as I've written it down before. One more important thing to note is I have to set center equal to false, so that because otherwise BRMS would center for me the covariates and essentially change the meaning of my priors, which I definitely do not want when I control exactly what's my prior. And then I give it as a prior this mix MV norm density with this syntax here. And this is a density which is not defined in stand, not defined in BRMS, but which RBEST will define for you by using this mix stand var link between BRMS and RBEST, which then defines in the stand code the respective density, which is just a straightforward implementation of a multivariate normal mixture in Cholesky parametrization. And it would also lead to adding these additional points which are needed here in order to define the density. And having all of that, then, does it read? Yeah. You can then do this statement, add prior plus equals mix MB normal PDF, and there all the data which we've just, or the mixture which we have fitted before will be passed into Stan, and we get our prior as we wanted to. So the results would be these. These are more accurate than not if you were not using the prior. So the study random effect has been dropped in this model as we've integrated it into the map prior itself. In this case, it's important to note that the covariate effects we have pooled across the different studies. So A for H and baseline score effect. So it's modeled without between trial heterogeneity. One could, of course, also try to do that, but then you would also have to write down the prior or give the prior for that and good rationale for it. So this functionality, I guess, is actually also useful in other settings, as I read quite often on the stand forums, how can I freeze my posterior, so to say, and this is actually a way to do it. So RBEST mixtures and BRMS are by now tightly integrated with one another. So RBEST mixtures, so the function to use is mix stand var in RBEST. With RBEST, you would fit your parametric mixture densities, and it has support for uni and multivariate normal mixtures. So especially the multivariate normal mixtures, of course, the one which you would go for if you have a more interesting model with more parameters. But you can also have it for beta or gamma mixtures, and then the RBEST mix fit uses expectation maximization, and it's heavily tuned for map priors as I have them in mind. So any funky multimodal density, I don't know what happens, we would have to see, no idea, but basically if you have nice clear peaks basically. Also it can deal with heavy tails because this is what map priors are typically. They're very heavy tail distributions because they are mixtures of normals, so something like a student T basically. There it actually works pretty well, and what's also always very sad when I present this, you don't see how fast it actually is. Really, you fit these and you basically get them immediately being estimated for you. Okay, this brings me to my end. Bayesian approaches are readily used by now for mostly earlier phase drug development stages. Formal Bayesian priors, where analysis is really Bayesian insofar that the prior sticks, you see require great care and use due to the many stakeholders. We document all the type 1 and type 2 error, as you've just seen, and write down in parametric form what our prior is, and so on and so on. Historic control data as presented, I'd summarize, is essentially a data-driven prior, which is why this thing is also so successful. Although the prior on tau is typically what sticks in your posterior if you want to. And really, yeah, good software like most standardized work is very critical to power these Bayesian analysis. for example, R-best, or particularly nowadays, actually, I work mostly with BRMS when I attack some problem. Okay, some acknowledgement before I close to my manager for basically supporting this, David Olsen, Beat and Heinz for working with me on the map approach over years, Lucas, Andrew and Björn for spending a ton of work onto the BAMDD website, Paul for his great BRMS package, and the Stan community for all those tools. Without this, it wouldn't have been possible. And with that, I'd like to thank you for your attention. Questions?
Yes. Well, first off, I think it's amazing that you can take people out of the control group and put them into the treatment group, because that's potentially life-changing for people. You know, you're at stage 3 of a trial, and these people can get better. So thank you. That's amazing. And then secondly, so as you're actually, I think that's it for now. Thank you.
So
I know.
we don't apply it for phase 3 typically. There it's like you should definitely collect the data just to be sure on that. But basically for phase 1 and phase 2, yeah, I mean you get faster trials being executed. If it's a disease where, yeah, people have a burden basically, and you have then the choice to go into 1-to-1 randomized versus the 4-to-1 randomized, which one would you pick? But on the other hand, I also always have to warn, is like the data you don't collect, you don't have. And with the map approach, it's great to basically make up for it to get and maintain power. But if there are other endpoints where you would also like to do comparisons, either you also do map there, have the data for that, or think about it, basically, again. Yeah?
So, what extent could you extend the hierarchy in the map and have tau itself be like an endpoint-specific parameter, like a exchangeability between different endpoints?
So with tau, we play some games, quote unquote, which is not for different endpoints actually, but what we do is we introduce sometimes different tau strata, for example, in order to weight the historical data. So for example, you could have a bunch of phase 3 studies which are highly homogeneous. You want to, or a few phase 3 studies which is highly homogeneous, or you want to put a prior on tau which reflects the homogeneity. It's important data, but you don't want to dismiss maybe other data reported in phase 2. But these are maybe more broadly including different patient populations, so you would put up a prior on that which reflects the greater heterogeneity, and then your map prior would be, of course, allocated to what you think, or what your data will be like. So this is what we do with tau.
I was wondering a bit more about the 4-to-1 ratio, because it seems like it's not. You said that I think the effective sample size of the prior was something like 25, but then the ratio was 24-6. So why wasn't it 20, or why was it like 31-6 or something like that? Like, what's the slip?
So first of all, the design as it had been run at the time just used a single beta. It wasn't actually robustified at all because the technique of robustification was introduced in 2015. And at the time, actually, the team was super confident that that's the prior to use, and we are very confident in it. And I think I tried it out. I think you can, not even sure if you can actually achieve, it's very hard to get I think you would need to see many non-responders on treatment. So the prior is really strong, actually, in this case, in all honesty. And I have to warn you a little bit. If the effective sample size is 25, it doesn't mean it's really like a sample size of, corresponds to a sample size of 25. This is like, if you're interested, we can go into it. It's the expected sample size, sample size this prior corresponds to. That's different. So in terms of the actual data which you're getting, it's different actually. So yeah, this is the plot to show there for this. So this is our data scenario. So if you have your trial, have your prior, and then you look like the different data scenarios. And let's say we put 6 patients on control, which is what we've done. So we have actually 7 different possible outcomes On the y-axis, you see marked the 36, which is the effective sample size of my map prior. And on the y-axis, we see the posterior ESS. So if I have one responder and response, that's basically what I basically would expect, then I get as a posterior ESS 45, which is more than +6. If I get something which I would actually not expect, which is like here something, then actually I'm in a confused state. This is what's happening. So the posterior ESS would be then below 36. And actually this is the prior predictive probability distribution as in terms of what you would expect, which outcomes to see given your map prior. So seeing 6 is something you would not expect.
Can you use this to understand like compliance? Like could you flip it and understand, oh, if you're finding something unexpected, maybe it's because the people are doing something unexpected.
In all honesty, I'm not aware of a case where really this, because this very unexpected case actually happens. The map priors typically, if you derive it properly, is a very good prediction of what will happen. But it can be, and this is where I do I have it? No, I don't have it here. This robustification bit kicks in basically. The robustification bit, if you add it to the to this plot here in the prior predictors, will give you like finite probabilities here that something unexpected can happen, basically. And then, yeah.
Yeah, I had a question around, first thanks for the talk. I had a question around sort of the linking back to frequentist statistics, because you showed the nice plots with like type 1 error and power. And I know that in informative metrics in general, there seems to be a bit of an obsession with identifiability of models.
Mm-hmm.
So if you have a system where you're like looking at osteoporosis and looking inside your bones, but you only have biomarker information on blood, if your model is not identifiable from that frequentist perspective, like what happens when you try a Bayesian approach?
What I've seen,
can it be made robust if you have simple priors?
I've seen if you start to use priors in pharmacometric models where you struggle with identifiability, they will help you a lot. It's just that the models will get, first of all, dependent obviously on your prior choices, so better be good in setting these and defending these, so be transparent about what you do there. Still, those models won't be simple to fit in Stan, obviously, but from what I've seen, Bayesian priors help you really a lot in these nonlinear models. So they become identifiable with Bayesian priors, but of course you need to be aware of that you've fixed the problem of not having the data by Bayesian priors, which is a choice to make.
Slightly less statistical question, but I read a thing a while back about it being used for rare disease research and using like previous medical records as historical data. I saw a thing in your slides at some point that said you only had sometimes like 3 previous trials that could be used to build historical data. Is there lots of work being done on what data is used as the historical data and how reliable it needs to be to build that prior?
Indirectly, yeah, of course. I mean, typically you would not have too much data, many data items in such a setting, and then basically the prior you put up on the between trial heterogeneity would reflect how much trust you put in it, in basically the previous data, how homogeneous it is, and it will basically then become part of the posterior by not being overwhelmed by the data, basically. So that's a way of weighting the historical data. The other way, of course, to deal with differences between trials is obviously with covariates, but with covariates it immediately gets more complicated in terms of, you need to model them basically, and then do you pool the covariate effect, do you not, and so on and so on. But still, for key covariates, I would certainly prefer to adjust for.
So is it previous trial that could you use a trial that had this method used as historical data in the future? Or does that not have worked?
No, sure, you could do that. Then I would basically rely on the just raw information and not the combine. In a way, you could then, if another trial has used the technique, you would be very lazy basically by just looking up their model and then use it again. Because after all, by the way, this map approach, if you don't robustify, it's actually right now as I have presented, it's a 2-step approach. You summarize some historical data in parametric form and then you use it as a prior. But it's completely equivalent to what we call meta-analytic combined approach, where you just basically jointly analyze the historical data and your new data, full stop. No need for this intermediate step of this parametric mixture, unless you want to robustify. The alternative to robustification is to use horseshoe priors or spike and slab priors, basically, which would be appropriate for that. But these are very unconventional in this domain, let's say, and that's why they are not yet used. Bob.
I want to channel for Andrew Gelman here and ask the question, so everybody's agonizing over the priors here. Why isn't anybody worried about the linearity assumptions in the model, right? You've got some big model that's making, are people like also worried about that and using something other than a simple GLM?
First, I've started with an intercept-only model. Nothing to worry about the big model here. And then, yes, I would actually only like to, I would only use these types of models when it's not a big model, actually. If it's very clear in terms of what parameter means what. And you are totally right, the linearity assumption is definitely there. I mean, in a way, as you've seen, I've shied away from it by making H categorical, basically. So, a way of nonlinear dealing with it. Yes, the baseline, the standard baseline was linearly in there, but this is basically like everyone would be doing this forever like this, so I would not challenge this basically way of doing it. So there's just basically certain ways of how to operate scientifically with these frequentist models, and basically this is where your center of gravity starts, and you take it from there and add the Bayesian bits basically. But yeah, I mean, right now the FDA actually has a Bayesian supplemental analysis call, which is there to ask sponsors to provide statistical analysis plans which would be augmented by a Bayesian analysis for, to actually just get to know and learn basically how for the health authority and for the sponsors to work with Bayes. So Bayes is really a bit in its infancy here, really, one has to say, because it's new insofar that you do have to have a dialogue with the regulators on what's your prior. And one example on the homepage from the FDA is also about, they have 3 examples. One is about, we want to have basically discuss about priors, so the log hazard ratio shouldn't reflect the prior on that the drug can kill the patient, and should also not reflect that the drug can cure the patient, basically. So this line of reasoning is basically asked for to have a conversation on with the health authority.
Thanks.
I'll ask. How much of your time is spent explaining this stuff to your biostatistics colleagues at Novartis?
At Novartis, I think we are a bit better probably because we are quite strong in all of this. But no, it's totally right. I mean, I've just explained you the hierarchical model. Come on, is that difficult? No. But is it difficult if you want to apply it and save patients' sample size? Maybe then you should really know what you're doing, basically. So, and nonetheless, often. Last year I gave a course on it, but I do explain it, of course, yes. And
I think we maybe have time for one more question. So if not,